Welcome, everyone. We're starting the Josep Carreras Distinguished Lectures of 2022. And we have the pleasure to have with us Dr. Benjamin Ebert. He's the chair of medical oncology at Dana-Farber and George and Jean Canellos Professor of Medicine at Harvard and a Howard Hughes investigator and also a member of the Broad Institute. In his previous training, he got a bachelor's degree from Williams College and a doctorate from Oxford University as a Rhodes Scholar in the lab of Sir Peter Radcliffe. He completed an MD at Harvard and a residency at Mass General Hospital and a fellowship in hematology oncology at Dana-Farber. His lab has been focusing on the molecular basis and treatment of pathological malignancies and also these pre-malignant, non-malignant conditions in MDS, myelodysplastic syndrome, and now in clonal hematopoiesis. He was the force, one of the main forces behind the introduction of thalidomide, where his lab showed that this is a drug that binds and to an ubiquitin ligase member and finally induce the degradation of proteins. And I think he will dwell on that in the lecture. He has found now many other analogs that are able to induce degradation in similar transcription factors. He has received many recognitions by this standing work. He's a member of the National Academy of Science, of the American Society of Clinical Investigation. He serves as president of the American Society for Clinical Investigation in 2017. And among the different prizes, he got the William Dameshek Award from the ASH, the Meyenburg Prize, the Schoben Prize from the Royal Swedish Academy, and the Stankos Meyer Award from the American Society of Clinical Investigation in 2021. So it's a great pleasure to have with us Professor Ebert, and the title of his talk is going to be Targeted Protein Degradation for the Treatment of Cancer. Please.
Thank you very much for that very nice introduction, and thank you for having me. So I'll talk today about targeted protein degradation for the treatment of cancer and these ideas of molecular glues, which is the cartoon on the right. These are my disclosures. Neomorph Therapeutics is a company that's interested in targeted protein degradation. So just as a very high level introduction that I'm sure you all know, how small molecules are different from degrader compounds. As you know, a large percentage of our drugs are inhibitors of enzymes. There are small molecules that bind to the active site of an enzyme and inhibit it. And to inhibit that enzyme, the drug needs to be present all the time, 24 hours a day, 7 days a week, and probably at a higher concentration. The drug needs to be at a higher concentration than the enzyme to make it fully inhibited. Degraders are a little different in that once a protein is degraded, it's gone forever until it's resynthesized, and so it has a longer lasting effect. And there are two broad flavors of degraders. There are heterobifunctional degraders, also known as PROTACs, and those molecules are quite large in general, and they have three components. They have a component that binds to an E3 ubiquitin ligase, a linker, and a component that binds to a protein target. These are very attractive in the sense that theoretically just about any protein that has a very specific ligand for it can be turned into a PROTAC and be degraded. The challenge of these molecules is that they are very large and they have these three components, and that all of them need to be optimized in order to get an effective degrader. Molecular glues are a little different in that they are small molecules, smaller than the bifunctional degraders. They rely on a broad protein-protein interface, and they provide just an extra bit of affinity between the protein target and the E3 ligase to induce protein degradation. And so these are harder to find because we need to identify protein-protein interfaces that are compatible, but they are very drug-like and can be very effective therapeutics. Either way, the E3 ligase polyubiquitinates a protein target and results in protein degradation. Now, the idea of gluing two proteins together would seem extraordinary and unlikely, but we've actually had such compounds for many, many years. Decades ago, Stuart Schreiber's lab showed that cyclosporin and FK506 act as molecular glues. These are both widely used therapeutics for immunosuppressive applications and transplantation and other settings. In the case of cyclosporin, it's a molecular glue between cyclophilin and calcineurin A and B. And in the case of FK506, a molecular glue between FKBP12 and calcineurin A and B. So these are widely used molecules. Natural cyclosporin is a natural product that have this very specific activity of causing an increased interaction between proteins. Now, I'll be talking the rest of the time about protein degradation. And so just a quick reminder of the E3 ligase system. There's 3 components, E1, E2, and E3 enzymes. There's just a couple E1 enzymes. And in these, the ubiquitin group is first charged onto the E1 enzyme in an ATP-dependent process. That ubiquitin group is then transferred to an E2 conjugating enzyme. And there's about 20 or 30 of those. And then E3 ligases are the enzymes that provide the specificity. They bind to particular substrate proteins and add ubiquitin groups. And in these systems, this is a cartoon of a modular E3 ligase, where the E2 binds, and for each ubiquitin, a ubiquitin group is transferred onto a protein substrate, and a new E2 binds and transfers its one ubiquitin over. So that's the system for polyubiquitination when there are K48 linked polyubiquitin chains that form on a substrate.Ose are recognized by the proteasome for protein degradation. So our entry into this field was through studying the thalidomide derivatives and their mechanism of action. And this is really a long story, many decades long, that began with the development of thalidomide in the 1950s in post-war Germany, where it was found that thalidomide caused decreased nausea with antiemetic properties and hypnotic properties. It caused sleepiness. And it was tested in preclinical models in rats and in mice, and it had no toxicity whatsoever, even at extraordinarily high doses. So it was thought to be an extremely safe drug, and was therefore used quite widely without further clinical trials, and even was used over the counter without a prescription throughout Europe, Canada, Australia, many, many countries. In the United States, in contrast, there was a young pharmacologist named Frances Kelsey, and she was one of her first jobs, and she said that she didn't think there was enough data to indicate that it was safe in pregnancy, and actually blocked it from approval. And in those days, pharmaceutical companies could just call up the FDA and say, what's the problem, and pressure them. But she blocked it, and during the time that she was blocking it, the first reports came out that children born to women who had taken thalidomide during pregnancy were being born at a higher rate with phocomelia, limba defects shown in this picture. And it took a little while because thalidomide was used so broadly, people didn't even think about telling the doctor they'd even taken it. But it was once recognized, quickly removed from the market. And in the United States, the modern FDA that governs our new cancer drugs and all other drugs really is based, many of the procedures are based on the experience of thalidomide, and the Frances Kelsey Prize remains one of the big prizes at the FDA every year. But thalidomide was continued to be studied in the laboratory. It was a very good TNF, it is a very good TNF alpha inhibitor, and has been used for the treatment of erythema nodosum related to leprosy. It has anti-angiogenic properties and was therefore tried in many different cancer types without efficacy, but in a trial of multiple myeloma, it had striking efficacy and ultimately was approved by the FDA for the treatment of multiple myeloma. Then relatively minor derivatives of thalidomide, lenalidomide and pomalidomide, had orders of magnitude better potency and efficacy for the treatment of multiple myeloma, and have actually become some of the most widely used drugs in cancer. The lenalidomide grosses on the order of $10 billion a year. So what an incredible arc from a drug that was one of the worst stories in the history of drug development to one of the top grossing drugs of any kind in the world. And lenalidomide has efficacy in multiple myeloma, as well as in myelodysplastic syndrome with deletion of chromosome 5q, which just happened to be the very subtype of MDS that my lab started to work on, and when my lab started, as well as some other B-cell lymphomas. But amazingly, all of this clinical development occurred without any understanding of the mechanism of action of the drugs. Lenalidomide acts, so we and other, Bill Kalin's lab and investigators at Celgene found that lenalidomide acts by binding to CRBN, and rather than inhibiting the activity of that E3 ligase, it actually modulates the substrate specificity. So lenalidomide induces a transcription factor, IKZF1, otherwise known as Ikaros, as well as its partner Iolose, or IKZF3, to bind to CRBN only in the presence of lenalidomide. So this interaction between CRBN and IKZF1 does not happen in the absence of drug. With the drug, it forms a ternary complex. CRBN is the substrate receptor for this E3 ubiquitin ligase complex. And once this full complex forms, lenalidomide induces rapid polyubiquitination of IKZF1 and subsequent proteasomal degradation. And IKZF1 and IKZF3 are master transcription factors for lymphoid cells, and myeloma cells require the ongoing expression of these transcription factors. And when they are degraded, lenalidomide and pomalidomide kill the myeloma cells. To give you an idea of the specificity of this, this is the very first proteomics experiment we performed. We've now performed many hundreds, but this is the entire proteome, a whole proteome study by mass spec in a myeloma cell line treated in 2 replicates with lenalidomide. And you can see just a handful of proteins out of the entire proteome change, including IKZF1 and IKZF3. So this is a very highly specific process that results in quite rapid degradation of these proteins. And you can see a dose response, you get an effect at 100 nanomolar or less, and it happens over a few hours. And this is pretty extraordinary because these are transcription factors, and we don't think of transcription factors generally as being easily drugged targets. They don't have active sites, they don't have easily identifiable drug binding pockets, but in the presence of lenalidomide, there is rapid and selective degradation of these transcription factors. Now, this didn't actually answer the question that we entered the field to address, which is why lenalidomide works in MDS with deletions of chromosome 5q. In further mass spec experiments, we found an additional substrate, CK1 alpha, that is selectively degraded. And CK1 alpha, it turns out to be one of the few genes that's always deleted in del5q MDS. And universally, and patients with del5q MDS have a deletion of one allele, a component of one allele of on chromosome 5q, but the other chromosome is entirely intact. So there is haploinsufficiency for a number of genes. CK1 alpha is actually an essential gene, so all cells will die if you degrade CK1 alpha entirely. And that led to the hypothesis that if there is 50% expression of CK1 alpha, further degradation will kill the cell, and a cell with 100% level of CK1 alpha will be relatively protected from degradation. That would give a therapeutic window for killing the MDS cells. So CK1 alpha is a bona fide substrate. This is an in vitro ubiquitination reaction where we can see CK1 alpha ubiquitination only in the presence of the E1, E2, and E3 enzymes and the drug. We now have crystal structure of drug-dependent binding of CK1 alpha to cereblon only in the presence of the drug. So it's a bona fide substrate of lenalidomide. Now, one of the next interesting twist in the story is proving this works in a mouse model. So we were really interested in proving that this worked. We made a CK1 alpha conditional knockout mouse, characterized it. And then the important experiment was to take CK1 alpha heterozygous knockout cells, deleting one allele, recapitulating del5q-MDS, treating with lenalidomide and showing that they have selective killing of the heterozygous knockout cells. We treated the mice. We got absolutely no effect. We also got absolutely no toxicity, even when we were treating with just crazy doses that the mice had no effects at all. We then realized that we should do an in vitro experiment before doing the mouse experiment, but we didn't. We took mouse cells. We'd always done our experiments in human cell lines, like normally we do. And when we treated mouse leukemia cells with lenalidomide, CK1 alpha didn't budge at all. We had absolutely no effect of the drug. We then realized that even in the 1950s, when thalidomide was tested, there was no effect of the drug in mice. And in fact, there is very little effect of these drugs at all, little or no effect in any efficacy studies in mice. So all these drugs were developed really based on clinical trial results in humans, not in vitro or not preclinical mouse models. Now that we know the mechanism, we can figure out why there's a species difference. We can see that in mouse cells that are treated with lenalidomide, you get no degradation of CK1 alpha. If you overexpress the cDNA for the mouse cereblon in a mouse cell, no degradation. If you overexpress the human cDNA for CRBN, the direct target of the drug, in a mouse cell, the drug works. You get drug-dependent degradation of CK1 alpha in the presence of lenalidomide. We systematically mutated the amino acids that are different between human and mouse and found a single amino acid change that's necessary and sufficient for drug-dependent degradation of CK1 alpha. So when you overexpress the mouse cDNA with the human residue at this one position, we get beautiful degradation of CK1 alpha. We then made a knock-in mouse that changes this one amino acid in the germline of the mouse. And now we have a model that responds to lenalidomide. This is a mass spec experiment, and we see no proteins that are degraded by lenalidomide in c-KIT positive hematopoietic progenitor cells in the wild type mice. But if in the homozygous knock-in mice, we can see that we get nice degradation of IKZF1 and CK1 alpha out of the whole proteome. Those are the top proteins that are degraded in these cells. Pomalidomide actually does not degrade CK1 alpha, and indeed it doesn't have efficacy in del5q-MDS, but it degrades another protein, ZFP91. And so you can see the knock-in mice degrade these proteins very well with pomalidomide. So we now finally have an in vivo model for studying these drugs. And this was a big field for decades of understanding why there was teratogenicity from thalidomide, and many organisms were tested for thalidomide teratogenicity, and primates have the human residue at this position, and except for one primate, the bush baby, which has the murine residue at that residue, and the bush baby actually does not have teratogenicity. Rabbits, in contrast, actually have the human residue at this position and do have thalidomide teratogenicity. So the thalidomide teratogenicity lines up perfectly with the valine or isoleucine at this position 391. In pregnancy, we've treated mice with lenalidomide, and the dominant effect we see so far is that there is pregnancy loss only in the knock-in, only when treated with lenalidomide. And the historical data from the 1950s would indicate that probably the dominant effect of thalidomide during pregnancy was pregnancy loss. It was generally taken during the first trimester for morning sickness, and so obviously pregnancy loss is fairly common and wasn't well documented because people weren't looking for it, but that's probably by far the most common effect of thalidomide. So if there were 10,000 babies born with thalidomide teratogenicity, there were countless more lost due to thalidomide. But we now know from collaborative work with Eric Fisher's lab that SAL4 is another transcription factor that is essential for limb bud development that is degraded by thalidomide, and that is likely, degradation of SAL4 is likely the cause of limb bud defects in response to thalidomide. So the next big question in the field was, what is the exact target sequence on proteins that's recognized in the presence of a drug to enable a substrate to be degraded? We did a saturation mutagenesis experiment and identified a single zinc finger that's in IKZF1 and IKZF3. It's actually same sequence in both proteins that mediates drug-dependent degradation. To do these and many other experiments I'll show you, we use a very useful reporter vector that Steve Elledge developed. It has an amino acid sequence cloned in frame with GFP, followed by an IRES and M-cherry. So if this sequence mediates drug-dependent degradation, GFP will be degraded because it's fused, and then the M-cherry will stay the same because it's a separate protein. So we can look for drug-dependent degradation by just GFP to M-cherry ratio by flow. So when we clone the full-length IKZF3 or ILOS into this vector, we get beautiful drug-dependent degradation. So lenalidomide and pomalidomide result in almost complete degradation of this reporter. So loss of GFP with retention of M-cherry. If we knock out CRBN, no degradation at all. If we knock out just zinc finger 2, no degradation at all. And then if we clone in just zinc finger 2 by itself, we get beautiful drug-dependent degradation. But if we mutate that zinc finger or knock out CRBN, no degradation at all. So just that zinc finger, a single zinc finger, is capable of mediating drug-dependent degradation of a reporter protein or heterologous protein. Working with Nico Tome's lab in Switzerland in Basel, we obtained a crystal structure of pomalidomide binding to just this zinc finger 2 of IKZF1 or IKZF3 in the presence of cereblon. And you can see that pomalidomide is indeed a molecular glue. It's interacting directly with CRBN in green and directly with IKZF1 in magenta. Plus, there are protein-protein interfaces that stabilize the ternary complex. And we can start to see this chemical groups that are distinct between the different compounds that mediate additional interactions that stabilize or destabilize the ternary complex and lead to the distinct activities of these different molecules. So if we can degrade a protein via a single zinc finger, one of the next questions is, are there other zinc finger proteins that could be degraded by thalidomide analogs? And that's of particular interest because zinc finger proteins are the largest class of putative transcription factors in the genome, and perhaps we could induce degradation of other zinc finger transcription factors that are of disease relevance that we've not been able to access pharmacologically in the past. So we cloned all zinc fingers in the proteome into this reporter vector, about 6,500. In fact, we've now done additional libraries that clone every adjacent pair of zinc fingers into similar vectors and continue to develop this library. But with this system, we can screen all zinc fingers, treat with various drugs, in this case, the established drugs, thalidomide, lenalidomide, and pomalidomide, and by flow cytometry determine which zinc fingers can mediate drug-dependent degradation. And the system worked beautifully. You can see, particularly with pomalidomide, out of all 6,500 zinc fingers, we rediscovered zinc finger 2 of IKZF1 and IKZF3 as one of the top hits, but we also found a whole bunch of other zinc finger proteins that have drug-induced degradation, and we've been able to validate many of them at the whole protein level, and follow them up. Additional derivatives, more novel derivatives, look like they induce degradation of new proteins and that might have novel pharmacologic properties. So what are, how do cells become resistant to thalidomide analogs? Obviously, cancer cells manage to become resistant to just about all of our therapies. What are the mechanisms to this sort of novel mechanism of action for drugs? So we and others have done whole genome CRISPR screens in myeloma cells in the presence of lenalidomide. These screens have worked really well. This is a screen with 6 guides per gene, 120,000 guides altogether, and hits 1, 2, 3, 4, 5, 6 were the 6 guides for CRBN. So out of the entire genome, hitting CRBN is by far the most effective way of blocking the activity of the drug and mediating drug resistance. So this is a very reproducible, many labs, many screens result. In addition, if you look at the top 30 hits, almost all of them are members of this ubiquitin ligase complex, regulators of that ubiquitin ligase complex, the E2 enzymes that bind to the E3 ligase, basically the entire biochemical system scores in the screen in the top hits. But CRBN is by far the top mediator of drug resistance. And that's been borne out in clinical data too. And so in general, we think that the mechanisms of resistance to lenalidomide as well as to other degrader proteins include mutations in CRBN or the substrate at the binding interface. So if there are mutations that prevent substrate binding, then the substrates aren't degraded and the cells can survive. There can actually be, in some cases, competition between substrates. So the ubiquitin ligase could be occupied degrading some other substrate if it's expressed at a very high level. We can demonstrate in the lab. That's hard to demonstrate in humans. And then probably the most common mechanism of resistance in myeloma patients is loss of CRBN expression. That happens through methylation and silencing of CRBN gene expression. It happens through genetic deletion of CRBN. It happens through inactivating mutations of CRBN, but that is the dominant mechanism of drug resistance. And the other consequence of this work is that, that each of the ubiquitin ligases that we can modulate therapeutically can be made into, can be used for heterobifunctional or degraders or PROTACs. So the 2 major types of PROTACs that are being used are molecules that bind to VHL or molecules that bind to CRBN. In both cases, those are joined by a linker to a target molecule. In this example, these are PROTACs targeting BRD4. And you can see that while I noted at the beginning that these have a linker and connect the 2 proteins via that linker, there's actually some protein-protein interactions that are going on, and the distinction between PROTAC and molecular glue may actually be not entirely distinct, because the most effective PROTACs likely have some stabilizing interaction, protein-protein interactions as well. So one of the big frontiers of this field now is finding new ubiquitin ligases that we can target pharmacologically to induce degradation of new classes of proteins. This is a huge field, as there are more than 500 E3 ligase components, many enzymes to target, many different substrates that they could potentially degrade. And so how can we start to identify those? Oops. One of the mechanisms that we've taken is a more bioinformatic one, where we looked for molecules that have activity that correlates with the expression of an E3 ligase component. To do this, we looked at the PRISM library, and PRISM is a huge data set that was developed at the Broad Institute. It's a platform in which 500 cell lines are cultured in pools that are with barcodes, and they're treated with drugs, here is a set of a data set with 4,500 drugs. So we have 4,500 drugs treated at dose response curves in 500 cell lines. So we have an IC50 for all 500 drugs. And we have RNA-seq data for all 500 cell lines. So we can look at the expression of genes in those cell lines and their correlation with sensitivity to all these drugs, and focusing on E3 ligase components. We found a bunch of correlations. The mass majority turned out to be false positives or not mechanisms of drug activity, but one of them, a drug called CR8, had activity that correlated with the expression of a protein called DDB1. And we could validate that CR8, which happens to be a CDK inhibitor, kills cells very potently, but when we knock down DDB1, cells are rescued. And that's actually kind of impressive because DDB1 is an essential gene and knockdown of DDB1 kills cells, but after brief knockdown, it actually rescues toxicity of CR8. So maybe DDB1 is involved in the in some activity of CR8 that mediates protein degradation. If so, DDB1 is an adapter protein. We don't know any of the other components of the system. So the first thing we did was mass spec, whole proteome mass spectrometry. We found that one protein, cyclin K, was particularly degraded after just a few hours of treatment. And if we do Western blots to validate, you can see really nice degradation of cyclin K after a few hours of treatment with CR8. But more importantly, if we add an E1 inhibitor, a netylation inhibitor, or a proteasome inhibitor, we completely block CR8 mediated degradation of cyclin K. Netylation is required for any Cullen ring ligase. And so these data would indicate there's a Cullen ring ligase that's mediating CR8 dependent degradation of cyclin K. So how do we find out the components of the system? We did genome-wide CRISPR screen for resistance to CR8. Since CR8 is a potent cytotoxic agent, we could look for what are the genes that are required for CR8 activity. We did a genome-wide screen. Again, DDB1 scored at the very top, validating the initial bioinformatic hit, but other components of DDB1 complex scored right at the very top. And that started to fill out this picture. We think that DDB1 is part of this E3 ligase Cullen ring ligase complex, including Cul4B, which scored, RBX1, which scored, DDB1, CR8, and cyclin K. But we don't have the substrate receptor. We don't have the part of the system that is required for recognizing cyclin K selectively. DDB1 is an adapter protein that doesn't seem to recognize substrates directly. So we did another screen. So there's a little delay here. And we did another screen, that this time a degradation screen. So we cloned cyclin K into a reporter vector in frame with DFP, genome-wide screen, and again found DDB1. This time we also found another protein called CDK12. That caught our eye because CR8 is a CDK12 inhibitor. We know that CR8 interacts with CDK12. We did not find any substrate receptor. So that raised the idea that maybe CDK12 was required for degradation, but there is no other substrate receptor. And that was proven by Nico Tome's lab and collaborating with him again, where they showed that if you just add CDK12, DDB1, Cul4B, RBX1, that complex, plus CR8, you can get polyubiquitination of cyclin K only in the presence of the drug with that complex without any other proteins. Moreover, they were able to solve a crystal structure of this, and it was surprising in that this is a molecular glue, just like lenalidomide, but it's not a molecular glue between E3 ligase and a substrate. It's instead an E3 ligase between an adapter protein, DDB1, and CDK12. CDK12 is not normally a part of an E3 ligase complex, but it becomes a substrate receptor, and then its partner, which is cyclin K, is degraded. So there's a very large protein-protein interface between DDB1 and CDK12, but those proteins don't interact physiologically as far as we've been able to tell in any system. However, with a little added affinity of CR8, which interacts with both proteins, they form a ternary complex. When CDK12 and DDB1 form a ternary complex, cyclin K binds as a natural binding partner of CDK12 and then is ubiquitinated and degraded. So to illustrate this in cartoon form, thalidomide and lenalidomide and pomalidomide are molecular glues between a substrate receptor and a substrate protein, or a neosubstrate protein, resulting in degradation of those proteins. CR8 is a molecular glue between DDB1 and adapter protein, and CDK12 becomes a neosubstrate receptor, and its partner cyclin K is ubiquitinated and degraded. Another really important consequence of this, or implication, is that when you compare CR8 to its parent compound, roscovatine, both roscovatine and CR8 are CDK inhibitors. They both bind to the ATP binding pocket of CDKs. And the only difference is this 2-pyridyl group outside of the ATP binding pocket that's on CR8. In a degradation assay for cyclin K, CR8 is an excellent degrader of cyclin K, but the roscovatine does not degrade cyclin K at all. So the only difference between these two molecules is this 2-pyridyl group, and that changes roscovatine into a degrader. So a non-degrader into a degrader just based on that change. And that 2-pyridyl group is not in the ATP binding pocket, it's solvent exposed right here. And that solvent exposed area is right where DDB1 binds. So that raises the possibility that an enzyme inhibitor that has already been identified could be modified with a group that is outside of the active site, but that would mediate an interaction with another protein, turn it into a molecular glue, and if that is a E3 ligase component, then that then it becomes a degrader. So at this point, there's actually multiple molecular glue degrader systems. The thalidomide one that we've discussed, Deepak Najwan and others identified indisilam and E7820 as molecules that act as a molecular glue between DCAF15 and RBM39, a splicing factor, resulting in cytotoxicity due to this molecular glue interaction and degradation of RBM39. And then this CR8, which acts as a molecular glue between DDB1 and CDK12. So all molecular glues, all resulting in protein degradation. Interesting, all of them part of these cullen ring ligases, which are these modular systems, including a cullen, the adapter protein DDB1, a ring finger RBX1 that interacts with the E2 enzymes, and in each case, either a substrate receptor, cereblon or DCAF15, or a neosubstrate receptor, CDK12. So we think that there are going to be more mechanisms like this, but we're trying to find even additional types of mechanisms of protein degradation. And one of the other ways of doing this is to go to the literature, and a number of molecules have already been reported to be degraders. One of these is a molecule that was identified by Boehringer Ingelheim. They have been interested for a while in targeting BCL6, a very potent lymphoma oncogene, and they were targeting the BTB domain of BCL6, which mediates a lot of protein-protein interactions. They identified inhibitors of BCL6, but they also identified one molecule that not only inhibits BCL6, but also degrades BCL6. And to their credit, they published the molecule, they published its structure, and that it was a degrader. They didn't have the mechanism of action worked out, but by publishing it, gave us and others an opportunity to try to work out the basis for the degrading activity. First thing that we did in collaboration with Eric Fisher's lab was to do whole proteome mass spectrometry. And you can see this molecule is incredibly specific. We see very nice degradation of BCL6 without degradation of any other protein. So very, very specific degradation of BCL6. And this inhibitor that was published as not being a degrader is indeed not a degrader. It doesn't cause any degradation of BCL6 at all. So relatively small difference between BI3802 and BI3812, the red and blue here, that changes an inhibitor into a degrader. So we did some mapping studies. We cloned BCL6 into our reporter vectors, started doing deletion mapping, and we could still see degradation of this shortened form of BCL6, but any deletions beyond the first 275 amino acids resulted in loss of the drug-dependent degradation. That's already a little bit surprising because we knew that the drug bound to the BTP binding pocket, but the deletion of some sequence that's removed from the drug-dependent, the drug binding pocket resulted in loss of degradation. So that's different from the glues that we had studied before. The other thing that was different and quite striking was that if we looked under the microscope at where the GFP was, after adding the molecule, we got these punctate foci that formed. And if we used a non-degradable form, the punctate foci would actually stick around until we would add BI3812, the inhibitor that was not a degrader, which would dissolve these foci and make them go away. So we've got a molecule that results in protein degradation very specifically, and that will result in foci of BCL6 to form quite rapidly within minutes of adding the drug. The BCL6 forms some sort of aggregates or co-localization in a cell. To understand the biochemistry, with Eric Fisher's lab, we made recombinant BCL6. And the first finding was that if we added this compound, BI3802, BCL6 would immediately precipitate. And you can see it at the bottom of the tube, and you can see it on this size exclusion chromatography that in pink, when you add the drug, you get higher molecular weight forms. So that could be just non-specific aggregation, or it could be some higher order structure that's formed. So to try to look at that, Ho-Jung Yoon and Eric's lab first did negative stain electron microscopy, and he saw this extraordinary picture where with DMSO, there are just monodispersed protein of BCL6 here, but when he added BI3802, you could start seeing these squiggles of that look like filaments of BCL6, helices, it looks like, of BCL6. And if you zoom in, you can actually get the pitch of these helices. He was then able to solve a cryo-EM structure, and amazingly, BI3802 is a molecular glue, but in this case, not a molecular glue between a protein and an E3 ligase, but a molecular glue between a protein and itself. So BCL6 acts as a homodimer. Each of these colors is a homodimer of BCL6, and BI3802, the small molecule, acts as a molecular glue between two homodimers of BCL6. Here in yellow and here in brown, resulting in these stacking and polymers of BCL6 that form, and as they get longer, they form this helix of BCL6. So we're getting drug-dependent polymerization of a transcription factor that ultimately results in highly selective protein degradation. To figure out why the protein was degraded, we did the same two genome-wide screens that I described before, a genome-wide degradation screen and a genome-wide resistance screen, and in both cases highlighted a ubiquitin ligase, CO1, that resulted that was required for degradation of BCL6, and its loss decreased the degradation of the drug-induced degradation of BCL6. And CO1, you can see in an in vitro ubiquitination reaction, accelerates the BCl6 polyubiquitination and then degradation. Exactly why that happens, we're still working out, but we think that it's an avidity effect. BCl6 actually does bind, CO1 does bind BCl6 even without the drug, but much less. And when BCl6 is polymerized, you get higher affinity or avidity of CO1 to BCl6 in ubiquitination of the BCl6 in this polymer, and then rapid degradation of the polymerized BCl6. And then I wanted to finally show one other application of this type of work, which is to use the degradation system as molecular switches. And we've done this in the setting of CAR T cells. So as you know, CAR T cells are extraordinarily effective therapy for a number of malignancies, particularly B-cell malignancies, but they are a live drug that are not in our control as physicians, and sometimes the T cells can become extremely activated, release high levels of inflammatory cytokines that lead to a cytokine storm and a cytokine release syndrome that can have significant morbidity and in some cases mortality. So could we have a way of controlling the CAR T cells if they become overactive? And this will be even more important as we develop CAR T cells for solid tumors. So lenalidomide is, of course, an FDA-approved drug. We know the toxicities, which are relatively minimal. And we can add a lenalidomide-responsive degron to a CAR construct. And in that model, once we add lenalidomide, the ubiquitin ligase would bind to the CAR construct, ubiquitinate, and then degrade. We first needed to show that this would actually work for a cell surface protein, but that was the idea. So if we have a CAR T cell that is killing a tumor cell, releasing a high level of inflammatory cytokines, we could add lenalidomide. The CAR construct would go away, but the T cell would remain alive. And as soon as we stop treating with lenalidomide, the CAR would come back and the activity of the CAR T cell would continue. So we've tried this both in cell lines and in primary T cells in vivo in human and mouse, and it works very well. You can see that if you treat with lenalidomide, the expression of the CAR goes away, and the activity of the CAR, as measured by release of IL-2, goes away with treatment of lenalidomide. To make them even more powerful, we did a variety of mutagenesis experiments and identified what we call a superdegron, shown in blue, which will induce degradation of the CAR at an order of magnitude lower concentration of lenalidomide. So at very, very low concentrations of lenalidomide, we can get degradation of the CAR and block the activity of the CAR T cell. So I wanted to close with just one final note of the difference in pharmacology between a degrader and an enzyme inhibitor. With an enzyme inhibitor, I mentioned at the beginning, the inhibitor needs to be present at high concentration to inhibit all of the enzyme. And anytime that the concentration of the drug goes below the therapeutic threshold, then the enzyme is active again, if oncogene or whatever is active again. In the case of a degrader, once a protein is degraded, it's gone for good and will be gone until the protein will be gone until it's resynthesized. And so the length of the effect is dependent on the resynthesis rate of the protein. In addition, the small molecule will induce degradation of a protein, but once that protein is polyubiquitinated and degraded, the small molecule is actually still there and can go bind to another protein and induce polyubiquitination and degradation. So you can actually get substoichiometric activity of a drug where the drug is present at a lower concentration than the target substrate, but the drug induces degradation of one substrate protein, will then go induce degradation of another and another. And so you can get activity at substoichiometric concentrations and long-lived activity. So these are the stories that I wanted to tell you about today, that thalidomide derivatives act as molecular glues. They induce ubiquitination and degradation of substrates by the CRL4 CRBN E3 ubiquitin ligase. They target a zinc finger degron, and different thalidomide derivatives target distinct substrates for degradation, and that determines their distinct clinical effects, both the efficacy and toxicities. For example, degradation of SAL4, which induces teratogenicity. In multiple myeloma, lenalidomide induces degradation of IKZF1 and IKZF3, and when that's essential for the activity of these drugs in myeloma, degradation of CK1 alpha is critical for the activity in del5Q MBS. And there's been a number of new molecular glue molecules that have been identified that result in protein degradation. CR8 is a molecular glue between DDB1 and CDK12 and induces ubiquitination and degradation of cyclin K. BI3802 induces polymerization and subsequent degradation of BCL6. And this is incredibly collaborative work, but I wanted to particularly note collaborations with Eric Fisher, who's an extraordinary colleague, structural biologist and biochemist at the Dana-Farber, Nico Tomei, who's also an extraordinary biochemist and structural biologist at FMI in Basel, worked closely with the proteomics group at the Broad Institute, and these recent stories have been led by Nikolai Slabicki and Max Jan, and I'm happy to take any questions.
Thank you very much, Dr. Ebert, for this comprehensive, but at the same time very focused lecture. So I like it very much. So remember that you can ask your questions using the chat in the web page there anytime. And I have some that already collected here. One is relates to the so why at the beginning was approved for multiple myeloma? What is so special about lenalidomide in multiple myeloma? You gave an example about MDS related to CK1 alpha. So
yeah,
there's something special about multiple myeloma that this drug worked so well there?
Yeah, it's a great question. So I'm not sure we would have known that a priori, but Icaros and Aiolos are essential for the survival of myeloma. They come out as dependencies in multiple myeloma cells and genome-wide screens. And these drugs were not developed as degraders of IKZF1 and IKZF3, but they are phenomenal degraders of IKZF1 and IKZF3. And those transcription factors have an amazing complementarity with CRBN. And so of the thalidomide analogs, new drugs keep being developed that degrade new substrates, but essentially all of them still degrade IKZF1 and IKZF3. They are the core substrates that are degraded very efficiently and very well because they interact so well with CRBN. So myeloma is the best malignancy to treat because they're most dependent on IKZF1 and 3, and that's just the pharmacologic happenstance of what proteins are best degraded by lenalidomide.
Thank you. There's another, you show in one slide a mutation in CRBN that distinguish primates versus rodents, more or less. That's one example of a small primate. So what this mutation generates for the protein? This activity?
Yeah, it's a great question. We don't think that there's any difference. So it may just be a random amino acid change during evolution. That actually brings up a broader point, which the challenge for us in the field is that we have not figured out what cereblon does endogenously. We don't know. It must be an E3 ligase, but we do not actually have definitively proven substrates of CRBN in the absence of a drug. We know it's important for brain development. If you knock out cereblon, that's why it's named cereblon, you get brain development defects, but major neurologic defects in the mouse. But we don't know what its endogenous substrates are. And if we did know, maybe we could answer that question better about whether that amino acid change subtly changes the substrate specificity of cereblon. I expect it would. And so maybe it's something important about the substrate specificity of cereblon, but we don't know the endogenous substrates, so we can't answer that yet.
Thanks. There is another here. It says, in addition to ubiquitination, there are other pathways involved in protein degradation, like the endoplasmic rat pathway, sumoylations. Some of this has been targeted using similar drugs.
So we don't have molecular glues for those other pathways, but there are really creative and clever ways of leveraging those other degradation pathways as therapeutics. Carolyn Bertozzi developed a system called LITAX, which uses the lysosomal pathway and grabs proteins with a ligand, pulls it into the lysosome and targets a protein for degradation. And that is an incredibly powerful approach that could potentially be used for the targeting of extracellular proteins or cytokines for degradation. So that's a novel space, and I think people are working on every mechanism of degradation to try to employ it to target proteins for degradation. We actually recently, or during the pandemic, we did a COVID-19 protein screen trying to find ways of targeting COVID proteins for degradation, and we did find that the envelope protein for COVID is targeted for degradation by an E3 ligase that's in the ERAD pathway in the endoplasmic reticulum. We haven't done a drug screen, but we postulate that that could be a mechanism of, if you could increase the affinity of the envelope protein for that ERAD E3 ligase, maybe you could degrade the envelope protein better and have a COVID therapeutic. So hopefully we could get better at those as well for all kinds of applications.
That's great. That's very original. I didn't think about that. That's very interesting. So thank you. I have another question here. It says, in the context of multiple myeloma, are also used proteasome inhibitors.
Yes.
And these drugs induce degradation. So how this works?
Yeah, that's a head scratcher and has been from the day one of this story. The relatively unsatisfying answer is that proteasome inhibitors and these degraders are both incredibly effective drugs for multiple myeloma. If you treat them at exactly the same time, you can completely block the activity of lenalidomide with a proteasome inhibitor. But the way that we treat multiple myeloma patients with infusion of bortezomib and then daily treatment of with a pill of lenalidomide, there's only a very brief time, if at all, that both proteins are on board, both drugs are on board at the same time. They're generally treating, they're not both at therapeutic concentrations at the same time. So they would nullify, proteasome inhibitors would nullify the effect of lenalidomide, but therapeutically we use them in a way that they're not both being used at exactly the same moment in the cells, and they're both just incredibly active drugs in multiple myeloma. And it's sort of strange how that happened, and we don't have another explanation other than that. But in vitro, you certainly can block the effect of lenalidomide with a proteasome inhibitor.
Okay. Thank you very much. In the interest of time, because we said that we'll close it in 1 hour, and we are there. So I would like to close here the lecture and the questions. Again, thanking the speaker for this great lecture. Thank you very much. Until next time, it has been a pleasure.
Thanks very much. Take care. Bye-bye.